Okay, so we don't want to be on YouTube.
No, but if you close that doesn't go.
Okay.
We're here.
Okay.
But
Okay. I think we'll be live in a few seconds.
Go back to the YouTube.
so this
Otherwise,
is,
it's going to do echo.
okay, so we don't want to be on YouTube.
No, but if you close that doesn't go.
So we're
Okay.
We're here.
Okay.
But
Okay. I think we'll be live in a few seconds.
so we don't want to be on YouTube.
No, otherwise it's going to do echo.
Okay, so we don't want to
Okay.
be on YouTube. Here.
Okay. No, but if you close that
Okay.
doesn't go.
I think we'll be live in a few
Here.
seconds.
Okay.
But
Okay. I think we'll be live in a few seconds.
Otherwise it's going to do echo.
okay, so we don't want to be on YouTube.
No, otherwise it's going to do echo.
Okay, so we don't want to
Okay.
be on YouTube. Here.
Okay. No, but if you close that
Okay.
doesn't go.
I think we'll be live in a few
Here.
seconds.
Okay. Okay.
That was an amazing little loop that just happened.
Exactly. Exactly. We start by apologizing.
Yeah,
We start
Cool.
by apologizing with these technological challenges. I think our audience is used to it. Sometimes these challenges happen while we keep streaming our talks. Good afternoon, everyone, and welcome back to the Primate Conversations series. We are very glad to have another amazing series of talks by a very diverse array of really brilliant researchers. And we couldn't start with a better example today. We are especially happy today to have with us Professor Robert Pringle for many reasons, because Rob does really research that is so interesting and so high profile, but also because, of course, our lab group, many of us work in Gorongosa National Park, and we follow with great interest the research from Rob's lab, as it really bears directly on our work on baboons and primate adaptations to complex environments. So we are all very much really looking forward to welcome Rob today. Just a brief introductory note, very brief. So Professor Robert Pringle is in the Department of Ecology and Evolutionary Biology at Princeton University, as many of you know. He has a PhD in biological science from Stanford University and 2 master degrees from Oxford, which I was very interested to find that you were here to do 2 masters. His research focuses on questions of species coexistence, the assembly of ecological communities, the organization of food webs, and patterns of biodiversity in space and time. He has quite a internationally known career, has published his work in top journals like Nature, PNAS, Proceedings of the Royal Society B, and so on, and has been featured multiple times in outlets like the New York Times, Washington Post, BBC. The list is very long: Scientific American, National Geographic, and other popular media. In Africa, Rob has done extensive field work, especially in Kenya and Mozambique. In Gorongosa, he and his team are doing what I think is really groundbreaking work and also essential work, focusing on the ecology of the large herbivores and the savanna restoration of Gorongosa National Park. And as I said, we are almost collaborators in a sense, because we are part of this large restoration project of Gorongosa. So we really look forward to this talk today, Rob. We thank you for accepting to join us, and we welcome you. Thank you, Rob.
thank you so much, Susanna, for that really wonderful introduction. And I do consider us collaborators. And I was going to say, actually, I would come back to Oxford and do a 3rd master's with you if you guys will have me. So when I agreed to give this talk, I didn't know exactly yet at that point what I wanted to talk about, and I figured it out last night. And and so this is the first path. I wanted to bring everybody to what I'm currently thinking about, and that, I think the two quotes that are up on the screen, which I hope I put up there just so people could look at them while we were getting started here, I think set the stage for what the theme, one of the main themes of the talk, which is to do with resource partitioning and coexistence in African savanna herbivore assemblages. So in other words, the challenge referred to by these quotes, and going back to G. Evelyn Hutchinson's seminal paper on the niche, in which he made this comment about the importance of understanding the biology of diverse herbivore assemblages while they can still be studied. The challenge that confronts a visitor to an African savanna is this kind of situation. This is a composite time-lapse camera trap photo by Jen Guyton, a former PhD student of mine, and it's showing 3 species of large herbivore, of antelopes, standing on the same termite mound. There they eat many of the same plants, and this poses a challenge for our understanding of coexistence. How can these species coexist despite the fact that they're clearly using the same habitat, eating the same food, and so on? At risk of oversimplifying, but only, I would say, by a little bit, there are 3 main mechanisms that are thought to be important for large savanna, large herbivore assemblages coexisting. One is that they differentiate along the grazer-browser spectrum, which is simply that some species, illustrated here by a zebra, eat grass. Some species here, exemplified by a dik-dik or a sunni, eat browse, which is to say non-grasses, all eudicots. And then some are sort of in the middle. They, like impala in the middle there, can eat grasses or they can eat eudicots, and they may switch seasonally or otherwise between the two. Then there's the idea that herbivores are, because they differ in body size and specifically height, they can use different parts of a plant. So a giraffe, a kudu, and a browser all have all have different maximum foraging heights. So giraffe can eat at the top of this shrub, a kudu can eat the foliage in the middle, and a small herbivore like a steenbok or a dik-dik can nibble shoots towards the very bottom of the plant, and in that way they can partition the foliage. And then there's the idea that the animals partition forage in time, and one classic manifestation of that is the Serengeti grazing succession, where different herbivore species move through the landscape at different times following the rain. So months after peak rain is correlated with growth of grass. So right after the rains, the grass is tall. Zebra are thought to come through and mow it down a little bit, and then wildebeest follow and they mow it down some more, and then Thompson's gazelles follow, and they are able to pluck daintily the smallest shoots. And in this way, one species is setting the table, I should say, for the subsequent ones. So all of these mechanisms and a few others have been known for decades, and numbers 1 and 2 up there are very well established. Number 3 is a little not as well understood, but it also seems that empirically it's not entirely clear that any of these 3 or all 3 together is really sufficient to sustain coexistence in herbivore assemblages. And there hasn't been a huge amount of recent, a lot of the recent work has been basically trying to consolidate our understanding of these mechanisms, and there hasn't been a lot of real progress towards linking these mechanisms to actual coexistence outcomes. So one of the problems, as was noted in that quote by G.E. Hutchinson, is that there are increasingly few places where it is possible to study such diverse assemblages of large mammals. another problem not mentioned by Hutchinson is that even in that subset of places where it is possible to work on these animals, we are focusing on a relatively small slice even of those. And I'm going to argue that there's much to be gained from expanding into places that historically haven't been researched as much. So there's a little digression here. This slide isn't really news, I think, to most people watching this, that the world's largest animal species are currently occupying a fraction of their historical ranges, shown for these maps in the blue, and with the current ranges in red for these three mega herbivores: elephant, hippo, and rhino. Nor will I think it surprise most of you that there are a series of factors that all of which contribute to the population decline and sometimes extirpation of these large animals. So this is from a review by Ripple and colleagues in 2015, and what they did to come up with the bar chart on the right here is basically add up the number of factors listed in the IUCN assessments as being a threat to a given species, and then they categorize them into 4 groups. But so on the one thing is that these are qualitative data essentially. Another is that there are interactions among these things that are not necessarily considered. So conflict can lead to runaway exploitation, among other potential interactions. And conflict, I highlighted here because it's something that we really don't understand very well for reasons that I will explain right now. So as to where conflict has happened in an African context, this is a map from a study led by a former PhD student of mine, Josh Daskin, and shows that all protected areas in Africa are the colored spaces, and they're colored according to the amount of conflict that's happened in those spaces since 1946, or rather between 1946 and 2010. And the shocking, or not maybe necessarily shocking, but discouraging, striking statistic is that 71% of African protected areas have had some conflict inside them in that period of post-World War II period, and 25% of them, a quarter, have had more than 9 conflict years. So conflict is really one of the things that we do know about conflict, is that it is widespread in some of the world's most biodiverse regions. And it is correlated with wildlife declines. This graph shows the annualized population growth rate on the y-axis, so basically the rate of increase of a population, lambda, plotted against the frequency of conflict in a protected area. And what you can see is that although there's a lot of noise in this relationship, primarily associated with the large range of possible outcomes during peacetime at 0 conflict, there is no population that was doing very well in an area with extremely high conflict frequency. And there is a clear negative correlation. So it's actually pretty neat that despite this widespread of populations, I should say also that the horizontal red line there corresponds to a population that is perfectly stable. And anything above 1.0 on the y-axis is a population that is increasing between 2 time points, and anything below 1 is a decreasing population. So at peacetime, populations can go extinct or they can proliferate wildly, but the mean is almost exactly replacement stability, right? And then that takes only a little bit of conflict to nudge that mean below the replacement level and into decline. So where does research happen? So one of the things that we noticed in compiling these data is that it's very easy to find wildlife population data from a few places, and you can guess which color those places are on this map. It's actually 80% of the wildlife population data that we were able to harvest in this intensive search were from 5 countries shown here: Kenya, Tanzania, Zambia, Zimbabwe, and South Africa. And the problem is actually even more pronounced than that, because within those countries, it's just a few protected areas where the most sort of wildlife population data come from. Ngorongoro, Kruger, Gunner Zoo, and a few others have an abundance of wildlife data. And then basically we know very little about what's happening in the rest of the continent, which impedes our understanding of sort of how conflict influences ecosystems. So this is a primate biology talk, and this is my one slide on primate ecology, which is basically habitat selection by researchers. And the data show that researchers select very strongly for bullet and landmine free habitat. Okay, so this talk, for the rest of it, is going to, well, almost all of the rest of it is going to focus on one place. It's a yellow place on this map, meaning that it has had a substantial amount of conflict in recent history, and that is Gorongosa National Park in Mozambique, a place with which many of you are intimately familiar. So the theme and the goals for today, many basic ecological problems remain incompletely solved, right? They've been pondered for many decades, and we have some answers, but not a fully, in to a large measure, the founding problems of ecology are still open, right? They haven't closed the book on very many of them. And that's because a lot of things are really intractable, and that includes large mammals at large scales. Perturbations, such as conflict, are useful potentially from a scientific perspective, even if they are tragic from a human perspective, because they act as natural experiments that can offer insights into problems that are not otherwise tractable, and that are difficult to study in fully intact and stable systems. So I also want to argue that by better understanding basic organizing principles, we're in a better position to facilitate recovery of perturbed systems. You have to know how the machine works before you can fix it. And the work that I'm going to talk about here today is part of a larger research program, and basically, focusing on many dimensions of the ecological and evolutionary dynamics of Gorongosa's recovery, but today really narrowing in on the one thing that I've been thinking about the most over the past few weeks and months, which is using Gorongosa in a comparative context to understand what structures food webs and what mechanisms underpin species coexistence in savannas. I'm going to try to show mostly new and or unpublished data. So this is the study system. This is a 4,000 square kilometer protected area in central Mozambique in southeastern Africa. And this is one of the ways that it looks from a certain angle. So this is savanna, where you have a mixture of trees and grasses. And what you can see going from the foreground to the background. So in the foreground, we have a relatively open savanna with not much tree cover. And those little clusters of trees with rings around them in the foreground are termite mounds. And termites build big mounds in this ecosystem. And on those mounds grow thickets of woody vegetation. In the mid-ground, sort of most of the way back in the photo, there's a belt of much thicker tree cover. And in the very far back, barely visible, depending on how big your screen is a vast open plain and a lake. That's the Urema floodplain, and so here's a view looking back in the other direction from where we just were, from standing near the lake in the floodplain, looking back at the transition, the boundary where the floodplain grades into savanna and woodland. So the defining feature of the history of Gorongosa, ecologically and sociologically in recent history, was the Mozambican Civil War. And this is a very long, complicated, terrible story that I'm not going to talk about. I'm just going to put on a single slide here to show you that it happened, because it's it's a pivotal piece of the context. In short, Mozambique fought and won independence in 1975, and then were immediately plunged into a civil war that lasted 15 years that was completely devastating to the country and to Gorongosa, where much of the conflict was fought. Everybody hunted, soldiers and civilians, and well, soldiers and civilians, which is a lot of people, and that persisted. There are some accounts of how this went down during the conflict, hunting on both sides by armies on both sides, and also after, when Mozambique was one of the poorest countries in the world. Millions of people had been displaced by the conflict, and people were hungry, and very understandably went and hunted their food. So these are photos taken in 1994, 2 years after the conflict ceased, and just showing some scenes of harvesting of wildlife. And the consequence of all this was the near total collapse of large mammalian herbivores and carnivores. So on this graph, we have the large herbivores broken down into two categories: mega herbivores, the big-bodied ones, elephant, hippo, and buffalo, and mesoherbivores, the medium-sized ones, antelopes and zebra. Their abundances, or their densities, I should say, per square kilometer in terms of biomass before the Civil War, so 1969 to 1972, from aerial counts conducted by Kenneth Tinley in the late 60s, early 70s. And then from a series of aerial counts, wildlife counts that happened immediately after the war, for the decade after the war, showing an almost complete devastation of large animal biomass. And the situation wasn't much better for carnivores. Lions persisted at low densities, but from left to right, leopard, spotted hyena, and African wild dog were all effectively extirpated, and remained so until very recently. Mozambique, as part of their long-term strategic plan, wanted to rehabilitate protected areas, and especially Gorongosa, their flagship protected area. And there were early efforts along these lines, but the progress accelerated dramatically in 2007 with the establishment of a public-private partnership to restore Gorongosa, and the signing of a long-term agreement to that effect. So this photo is then President Guebuza at the ceremony establishing a multi-decade agreement between the Mozambican government and the Carr Foundation, which is a US-based NGO, for the co-management of Gorongosa National Park. And that deal and the subsequent tremendous effort by many, many people and organizations has enabled a very impressive recovery of Gorongosa. So much so that I first visited Gorongosa in 2010, and at that point it was something that not many people had heard of. And by now, I think it's safe to say it's famous worldwide for being an example of recovery, ecological recovery and resilience. And this graph shows the recovery of large herbivores. So continuing the time series in that bar chart that I started to show you before. But you'll notice that there's been a flip in relative abundance. So whereas before the war, the largest bodied herbivores were the most abundant, now it's the medium-sized ones, and a few in particular, that account for by far the largest share of biomass. As for carnivores, the lion population has recovered substantially. The African wild dogs were introduced starting in 2018, and I believe are now more than 100 individuals. And leopards have just been reintroduced over the last year, so probably fewer than 10 total in the whole park, but that number is expected to grow. Hyenas, not yet, but within the year is the plan that they will be introduced. Here are the recovery trends broken down by species, and just to emphasize a few, particularly two, waterbuck and bushbuck there on the bottom right, and more broadly, this set of 5, which, as you can see from the y-axis on these plots, are the most abundant species in the system. So whereas wildebeest are, still at a small fraction, just for example, of their pre-war abundance and at very low overall densities at 0.25 individuals per square kilometer. Waterbuck are on an exponential trend upwards with many individuals per square kilometer, and some of these other species are similarly quite abundant. So we study this dynamic and very obviously non-equilibrial system for insights into what's normal and what is odd compared to intact systems where almost all of the literature is based. And again, I'm going to omit or gloss over a lot of the details of the research program and focus on one particular piece of it. And for that, first, I want to introduce 2 main tools. One is a technique for diet analysis, DNA metabarcoding, and one is a statistical approach for making sense of multidimensional data, which is various kinds of ordinations. And so I'm going to start showing ordination plots. I'm not going to talk about the statistics, just wanted to forewarn you that there's going to be a lot of ordinations. So the tool is this. For diet analysis, we start with a fecal sample, and then we take the undigested plant cells in that fecal sample, sequence the plant DNA, and match it to a local library of reference DNA from plants from Gorongosa, and in that way we characterize the diets of herbivore and other animal species. It's a very powerful tool, and also has the advantage of enabling us to count DNA sequences, which provides a reasonable first approximation for the amount of each food, in addition to being able to identify them precisely. Some people ask why we have to go to that trouble, and the answer, the short answer, is in these photos. And I would challenge anybody who's watching to tell me what plant species each of these animals is eating, and I don't think you can. And even if you can, I don't think you could, even if I zoomed in on these photos. Even if you could, you wouldn't be able to tell me, even if you watched these animals in real time, how much of each plant species they were eating. So just to underscore this in a slightly more quantitative way, these are data from this survey, this pre-war survey of Gorongosa, Tinley's work that I mentioned earlier, showing he had feeding observations for 11 herbivore species that over 5 years noticed that they collectively ate 181 plant taxa. We collected fecal samples from the same 11 herbivore species in just one dry season, and our network looks like this. So this is, again, yellow points are herbivores, blue are food plant species. And the short story, the difference between these 2 plots is that they detect a similar number of food plants. So we detected 151 food plants, but 25% more total interactions, or that is to say links between herbivores and plants, and with the ability to weight links by the amount of plant DNA present in the samples. Okay, so this is a really, I put this in here to underscore what a lot of people don't realize, and I think is even more pronounced for large herbivores, which eat foliage often of stubbly plants on the ground, than it is for, say, primates, where people have had more success in following and watching. You can, or for insects for that matter, where you can actually see the animal on the plant that it's eating. A mouthful of food for an elephant can comprise a dozen plant species that are growing together in a clump. So figuring out diet is actually a bigger challenge than people have long realized. Okay, so here's our first ordination. This is a non-metric multidimensional scaling plot. And the only thing you really have to know about this is that points on this graph that are closer together are more similar in terms of their diet composition. So each point is a fecal sample. Points that are similar have very similar diets. Points that are far apart have very different diets. And this graph shows something that will become clearer in a second, which is that the herbivore species in Gorongosa are very messy. Let's say they're messy eaters, but they are messy in their sharing, in their partitioning of resources. And you see that by virtue of the fact that there's a giant cluster of points sitting on top of each other in this graph. So just to orient you slightly, I've got the species names by color on the right, but all right, so I'm going to put these ellipses on this graph just to add a little bit of order to, but not much, to the chaos. And to orient you here, the species on the right and the top right are grazers, the grass eaters, and the species on the lower left are browsers, the non-grass eaters. And I want to draw your attention to, again, to the two species that I starred in the earlier graph. So one is waterbuck here, this very, very abundant, rapidly increasing species. And it's a little hard to see with all the clutter, but the orange points signifying waterbuck diets are distributed all over this plot, all over the sort of the upper, this upper half above the diagonal. So indicating that they're mostly grazers, but really they're eating all their diets are all over the map here. And here are bushbuck, the pink points, which also span along a large distance along the y-axis. So just keep that in mind. I'm going to come back to it. The real point of this graph is how messy and ugly the pattern is, or the lack of pattern is, rather. You can see that some species do separate, like for example, on the species on the left, the browsers, separating more than the grazers, but by and large, it's a mess. And that stands out in particular in contrast to data that we've compiled using the exact same methods in 10 different ecosystems, spanning from just north of the equator in Laikipia, Kenya, all the way down to the tip of South Africa in Addo National Park. And what we see everywhere we go, except Gorongosa, is much starker lines of food partitioning, where each species basically forms a little cluster of points. When we sequence the plant DNA from each species, samples are more similar to each other within species than between species. This is a very general pattern we are finding, we have found, haven't yet published. These are data that are in a manuscript that's still in revision at this point, and that I've been working on, which is why I wanted to talk about this stuff. And again, I'm just sort of, all right, there Gorongosa goes blinking to remind you what it looks like, and how it contrasts with these much cleaner lines, more modern, cleaner lines in these other assemblages all across Africa. And I'm not going to go through each one of them and go through what each species is. The species are listed there on the right. They don't really matter. The point is that we have clusters of species segregating together everywhere pretty much but Gorongosa. So a hypothesis relating to the interplay of herbivore and plant traits and the interplay of top-down, that is to say predation, and bottom-up, which is to say resource limitation and competition forces. So the general pattern, here's a hypothesis, the pattern of clean food plant partitioning that we see throughout Africa stabilizes herbivore diversity by decreasing interspecific competition and increasing intraspecific competition relative to, right? So by increasing the likelihood that a species is limiting itself rather than being limited by other species, which is a recipe for competitive exclusion. And this occurs because interspecific competition causes each species to eat plants with traits that species is best able to harvest and process, right? An elephant is able to reach and process taller and tougher foliage, right? So maybe elephants are the only ones that eat palm trees in Gorongosa. Indeed they are. by the way. So there's a trait matching between herbivores and plants. Secondly, because predation risk causes species to segregate in space according to their own vulnerabilities. So an elephant is not very vulnerable to predation, whereas a small-bodied animal like a bushbuck is. And different species have different strategies. Some hide to avoid predators, such as bushbuck. Some run, such as wildebeest, zebra, impala. And those differences should lead to species also favoring areas where they feel safe. So this broad hypothesis leads to some specific predictions. One is that plant traits differentiate herbivore diets, and I expect to see this even in Gorongosa, where interspecific interactions are weak, we believe. Herbivore traits differentiate diets, and again, we should see some signal of this, even if muted, when interspecific interactions are weak. But then release from interspecific limitation, from limitation from release from interspecific competition and predation should lead to intraspecific niche expansion. So if I suddenly don't have to worry about being safe, I can go anywhere and I can eat the plants that are growing anywhere. Similarly, if my population becomes very high, I'm going to search for ways to differentiate myself from my conspecifics by eating a different diet. In this way, competition within species can lead to expansion of the dietary niche and individual variation within species. And that should lead to this pattern of overlapping dietary niches that we see in Gorongosa. And last, and I'm going to tell you right now, I don't have the answer to this one yet, but it's still a prediction, is that restoration of these interactions in Gorongosa will, with time, lead back to the general clean pattern. So, with respect to prediction one, indeed, plant traits differentiate herbivore diets. So this is a study just recently accepted by my PhD student, Arjun Potter. A fabulous study that I won't be able to do justice here, but really, really cool work. And what this is a linear discriminant analysis. So it's an analysis that identifies linear combinations of variables that that maximize the separability of known classes. So it's basically saying which plant traits best predict the species identity of a herbivore diet. And the vectors here are associated with traits, which are the text. So tensile strength is a plant trait, and as I said, elephants are most likely to be able to eat really tough leaves like palm leaves, and you can see that tensile strength arrow is pointing up towards the yellow dots that correspond to herbivore fecal samples. So this is basically mapping plant traits onto herbivore diets, and showing which plant traits are associated with which herbivore species diets. And there are some very strong patterns, and it may be a little hard to digest in this this many-dimensional format here. So I'm going to try to simplify it. So this note here that there's separation of herbivore species along the horizontal axis, linear discriminant 1, LD1, and also along the vertical axis, LD2. So the separation that we see along LD1 is explained almost entirely by the percent grass in the diet. And LD1 indeed corresponds to traits that differ between grasses and non-grasses. So hemicellulose is higher, leaf thickness is lower, various other traits. So that separation along the horizontal axis of that plot is just grazers versus browsers, but in terms of the plant traits that differ between grasses and dicots. LD2, I think, is more interesting, and this is traits that separate herbivores according to their size, their body size, and I should said each point on the upper grass is again a diet sample, and here we've pulled out their scores along each of these axes and plotted them against biological variables. So on the right, in the upper right, it is each point score on LD2, showing that it's correlated with herbivore body mass and also differs between these two main Gorongosa habitat types. So linear discriminant 2 is some measure that combines the effects of body size and habitat use. And the traits that are important there are things like tensile strength, body size, zinc content, interestingly, sodium content, interestingly. These are traits that haven't really received much attention in context of large mammalian herbivore diets. So I want to sort of pick up on this thread that size and habitat are body size and habitat use are differentiating diet, and see if we can illustrate how that might occur. And to do that, I'm going to tell you about some work we're doing on 3 congeneric antelope species, either the spiral horn antelopes, Trigelaphus or Trigelaphus, and they include the 3 species shown here: bushbuck, the smallest at 33 kilograms female body weight, Nyala, intermediate at 73 kilograms, and kudu, the largest, at 120 kilograms. And you notice they're all standing here on this big termite mound. And there's a bushbuck there at the top, browsing some foliage there. Termite mounds are, as you saw in that earlier photograph, hotspots of woody vegetation for browsing antelopes to eat. So the first thing we notice is that, all right, so our project, we call it Ashamed. It's the allometry of spiral horn antelopes, movement ecology, and diet. And we find that space use scales allometrically with body size, right? In these congeneric browsers, we're controlling for the effects of phylogeny. And we see that homorin size increases as a nonlinear function of body size, which is generally the case in mammals. We find that selection for termite mounds, which are these localized hotspots of high quality concentrated foliage, decreases allometrically, nonlinearly with body size. And this graph gives you a way of visualizing that. So on the y-axis, you have the distance to the nearest termite mound, and then each point on these graphs shows you the number of times that that one of these animals has revisited that site, depending on how far it is from the termite mound. And what you see is that bushbuck, all the points are clustered pretty close to termite mounds. Bushbuck rarely venture far from termite mounds. Nyala venture a bit farther, and kudu venture the farthest. But they all significantly select for these hotspots, just to differing strengths. Those differing strengths translate into different selectivity for plant species that differ in the strength of their association with termite mounds. This is complicated. There's a lot of data here. I hope it makes sense, but we'll have Q&A if it doesn't. So each of the pie graphs here is a plant species, and the shaded part of the pie is the proportion of that species' total abundance that exists on termite mounds. So Cleistoclamus kirkii, the far left plant, occurs on termite mounds, but most of its abundance is elsewhere. Burkemia discolor is exclusively found on termite mounds. It is a termite mound specialist, right? And so forth for the rest of these 8 plant species. And the bars here show the selectivity, so the use relative to availability of each of these plant species. And without going into super detail, just looking, for example, at the first three plant species on the from left to right, you see that bushbuck, nyala, and kudu differ in the degree to which they're selecting these different plants. Kudu, which range more widely from termite mounds, are strongly selecting the ones that grow least on termite mounds, that are most widespread off of termite mounds. Bushbuck and nyala select strongly for plants that occur only on termite mounds, and so on and so forth. So there's some congruent patterns in their selectivity, but also some differences between species that reflect their differing space use, and those differences lead to differences in diet composition and quality. So on the left is another NMDS, non-metric multidimensional scaling ordination, where again, closeness indicates similarity and farness indicates dissimilarity. And it's showing that across years, these 3 species eat significantly different diets, right? So they're kind of bucking the general Gorongosa pattern, but conforming to the larger pattern out there. And this emerges from body size dependent variation in the use of space. And I'm going to skip over, in the interest of time, the graphs on the right. Suffice to say, these differences in diet composition also translate into differences in diet quality, because I want to move on to my third prediction, which is about release from interspecific interactions and what that effect that has on the dietary niche. So the waterbuck population in Gorongosa has grown logistically, which is shown here. Basically, the actual data points from wildlife counts are in red, and then the green interval and red line is the prediction of a simple logistic growth model that, with different assumptions about carrying capacity. And waterbuck conform beautifully to the sort of intermediate carrying capacity assumption. And what logistic growth means is that you're not limited by anything but yourself. Your own intraspecific density dependence is regulating you, and you are not being suppressed appreciably by competition from other species or by predation. This population explosion with waterbuck, has led to, so in 2018, there were 81 waterbuck per square kilometer in the floodplain in Gorongosa. That's 16,000 square, on the order of 16,000 kilograms per square kilometer. Sorry, that's a typo on the slide. 16,000 kilograms per square kilometer in their preferred habitat. And as you can see in the little graph in the upper right, as density got very high in the floodplain, and as we showed that they started to deplete their preferred food plants, individuals moved out of the floodplain into savanna. And you can see that by the massive yellow in the bottom graphs, which just shows where animals were in aerial counts. And basically the answer is they were everywhere. Saturating space. These are just movement data from individual collared animals showing, again, that they're happy in the floodplain, but that they're also moving into the woodland. And with that habitat expansion comes diet expansion within species, right? So this is just the same set of graphs repeated over 4 years. And on the left of each one is a ordination, so dissimilarity, showing savanna and floodplain and savanna woodland diets differ. And on the right is bar graph showing which plant families they ate more or less of. But the point is really that they differ, and because they differ, the species, the population level diet has expanded to include this whole set of savanna plants that historically weren't in the diet, right? So that explains this problem, that waterbuck are they're literally all over the place, and so are their diets, and overlapping with everybody else in the process. So the last little piece of this story is bushbuck, which are doing something very similar, but for a different reason. So what we know about bushbuck is, or what we thought we knew, is that it's what it says here. This is the standard safari guide. It's a colorful antelope. That's true. It lives in patches of woody vegetation, and wherever there is enough cover to conceal it, it is dependent, a forest edge antelope dependent on concealment to avoid predators. And yet, we started to find this. We were kind of a chance discovery happened in the course of trying to put collars on animals and getting frustrated because we were having a bad morning, and we took a drive out onto the floodplain and noticed there were a lot of chubby bushbuck hanging around, looking like they were really plump and happy and were easy to collar. So we started digging into this. What are these bushbuck doing out in the middle of the open? That's very unusual. And indeed, if we go back and look at aerial counts, we see that bushbuck have been steadily, increasingly expanding. The floodplain, by 2016, the density of bushbuck in the floodplain was higher than it was in the traditional and historical, in Gorongosa, woodland habitat. So like waterbuck expanding into the bush, bushbuck are expanding towards the water. These are home ranges from individual collared animals, where the orange are individuals occupying the floodplain and the blue are individuals occupying the conventional habitat, woodland. And just like in waterbuck, they eat totally different diets, again leading to intraspecific niche expansion through individual variation. So on this graph, this is yet another ordination plot of dissimilarities. Here, each point is a fecal sample and each polygon is an individual that was sampled many times. So Hallie Walker heroically managed to, well, it may not seem heroic to people who work on primates, but it was heroic to us. These bushbuck are a little bit shy, and Hallie was managed to get close enough to our collared animals to be able to repeatedly sample the same individuals up to 15 times, giving us some sense of repeatable individual variation. But the main point of the slide for present purposes is that the woodland and floodplain individuals' diets are radically different, except for a couple bushbuck who crossed over, moved habitats. So again, habitat expansion leads to dietary niche expansion. In short, waterbuck and bushbuck have expanded out of their historically preferred habitat in opposite directions and for different reasons. For waterbuck, it was intraspecific competition, density-dependent limitation, depletion of food resources in the floodplain, causing them to move into the savanna, where they actually have a crummier existence. They're more parasitized. The plants are worse in quality, so it's not a place they want to be, but they have to, because the intraspecific competition is very strong in the floodplain. For bushbuck, the trigger was relaxation of predation risk, and I didn't tell you how we know that, but there's a paper about it at Atkins et al. 2019. And individuals moved into the floodplain and ate high-quality diets. So they're exploiting an opportunity created by the low predation risk prior to 2018, resulting from the extirpation of Gorongosa's carnivores. In both cases, intraspecific niche expansion leads to the trophic mess that we see in Gorongosa, all of this overlap in diet composition, which I expect to be transient. So prediction 4, that continuing recovery of Gorongosa will lead the graph in the top right, Gorongosa's herbivore niche partitioning, to match much more closely those shown elsewhere on this slide, where each herbivore species eats a distinctive suite of plant species in distinctive relative abundances. But that, unfortunately, my story ends here because we don't know yet. We're continuing to monitor this. So I'm gonna, in the interest of time, I had one last piece in case I had time to get to it, but I don't, about the functional implications of all this. But I want to skip to my conclusions. So number one is that in studying a heavily perturbed, recovering ecosystem in a comparative context gives us general insights into the forces that structure African savannas. So we found evidence that large herbivores partition food resources based on plant and herbivore traits. And this is shedding light on coexistence mechanisms that are general, that we observe in Gorongosa despite the historical shakeup in community composition. Yet we also find that the patterns of resource partitioning are much stronger everywhere else but Gorongosa. And by digging into the biology of some of the key and abundant species in Gorongosa, we're able to actually tease apart the factors that are causing this. Like, why are the bushbuck and waterbuck diets so broad and therefore so much overlapping with those of other species? It's a result of a breakdown in the species interactions. So species interactions are the key to maintaining these clean patterns that we see elsewhere in intact systems, is the inference that I draw. And lastly, the anomalies of Gorongosa are likely to dissipate as the recovery continues. This was a conclusion based on data that I didn't have time to show you, so I'll skip it and go right to my acknowledgments. And this is many, but not all, of the people who have contributed and in most cases led the work that I talked about today, and tremendously grateful to them, as well as, of course, to Gorongosa National Park. and the government of Mozambique for allowing us to work there. And particularly want to highlight Dr. Mark Stallman's in the bottom row there, whose leadership of the Scientific Services Department in Gorongosa has been have made it a true pleasure to work there for these last 12 years this year. So I'll stop there.
Thank you, Rob. Really brilliant talk. Gives us a lot to think about. And we do have several questions from the audience, if I may.
Please.
We have a set of questions here, questions that are related from, let's see, from Joanna and from Yana. And these have to do with the few species that have not recovered well. So you show the zebra and wildebeest. What is it about their biology or their ecology that, unlike the other species that you've shown, has sort of held them back as others recover?
That's a great question. And the short answer is I don't know, but like any academic, I'm not going to let that stop me from giving my opinion. Well, all right, so let me actually back up a bit. So we don't know, and this, so I would also put eland in that category. The 2020 aerial count, they counted about 70 eland, but only 28 in the main count block. So eland also is sort of a low abundance species. And this summer, this northern summer, June, we're going to try to put collars on zebra and eland. We basically have very few data on these species because, as many of you know, finding them is extremely difficult. So we have, some sense of their diet, but no real sense of why they're not recovering. But we're very interested in that. What I want to show is this slide, which is from this is a really striking paper by Gutu et al. In 2012 about Lake Nakuru National Park in Kenya. And what I want to draw attention to, if I can, I don't know if people can see my mouse here. No, you can. Okay, great. Is waterbuck and warthog. These are two species very abundant in Gorongosa, and look what they did. They peaked. So this is also an ecosystem with a lake, and it's kind of like Gorongosa in some respects. Not many, but some. It's like it in that it has a lake. And warthog and waterbuck peak and then crash, right? Zebra, buffalo, increase steadily, right? And so sometime in the early, I guess it's the, I don't know when exactly it was, maybe early 2000s. At some point in this trajectory, zebra and buffalo displaced waterbuck. So I think waterbuck is a poor interspecific competitor. Something it's not obvious to me why it is prone to explode the way it has in Gorongosa and in elsewhere, as this paper showed. But they do seem to be crummy interspecific competitors. They have very high water and dietary protein requirements. And what we concluded based on looking at their diets in floodplain and savanna is that they're not going to hack it in savanna in competition with savanna adapted species. And so what I suspect is that, right, over time, we should see this. Now, the real question with zebra in particular is, are they in some kind of, are they at some critically low density that they can't escape, that they're failing to punch out of because of some kind of demographic stochasticity or something like that? And that's what we hope to discover by learning a little bit more about them. But in general, this is sort of like my mental model for, what the 40 to 50 year trajectory of Gorongosa might look like. But I'd love to know, we're going to hope to know more about the details of whether zebra are actually increasing, and it's just the very early stages. When you start with 12 or 20 individuals, right, it can take quite a while before that exponential or that first phase of growth picks up.
Mm-hmm.
And Rob, just to follow up on that same question, we have a question here about disturbance in the Lake Nakuru area. Was there anything comparable there that might be like what we've seen in Gorongosa, or is it a very different context?
Very different, but similar. So what happened was they took a small park and expanded it. So suddenly the densities were very, very small. because they appended a bunch of adjacent properties to make the park bigger. So effectively, it was an empty place, but not because they were abundant and people eliminated them, but rather because they started from a small population in a small area that when you basically massively increase the denominator by making the park bigger.
Thank you, Rob. I think that's, yeah, the zebra puzzle is definitely one that we all think about when we are out in Gorongosa. We have another question from our audience asking, what timescale do you predict is needed to collect data to establish food webs and species coexistence in a place like Gorongosa, especially with such variation in recovery across species?
Yeah, that's a really good question. So a great asset of metabarcoding is that we can gather a lot of data on diets very, very quickly. In the past 5 years, I don't know how many thousands of fecal samples we have, but it's several. But that's, so in terms of getting data on sort of how quickly this dietary niche partitioning picture snaps into place, that's not hard. The really hard thing that the question gets at is actually linking that to coexistence, right? And that's not trivial, and nobody's ever done it. So all of these mechanisms, mechanisms of resource partitioning are stabilizing mechanisms that stabilize coexistence, but they don't show that in themselves, and they cannot show in themselves, that intraspecific limitation is stronger than interspecific limitation, which is the criterion for coexistence. And that, so what the time scale is there, I don't know, because I don't know exactly how to do it yet, although we're thinking about it a lot. But I do think, and apropos of zebra, that Gorongosa and other, maybe even more so, other rewilding or species translocation scenarios are opportunities to get a better, more, to actually make this connection. So with zebra, if they do not increase from low abundance, by definition, they are not coexisting, right? If zebras stay at 30 individuals, I don't care how long, they are not coexisting, because the definition of coexistence is the ability to increase from low abundance in established community, right? So by studying zebra and Gorongosa, we may actually get some insight into that, but I can't say what it might be yet because we haven't done it. But other translocations where, for example, you may have an established community, let's say Gorongosa's carnivore community, when, let's say it's this year and hyenas are going to be reintroduced, and we already have established populations of lion and wild dog. Those kind of situations where you're introducing a new species into an established community are a strict test of coexistence, because any species that is translocated, establishes, and increases is by definition coexisting. And then if there are niche shifts in the established species resulting from that, then we can infer, I think safely, that those niche axes are important actually mechanistically for coexistence. So my wheels are turning about this stuff too. But I think that, again, it comes back to the theme of the talk, and the theme of the talk comes back to the paper that I'm trying to write, which is on this, that these kinds of rewilding scenarios offer us really tremendous and I think underused, scarcely used opportunities to get at things like this, that are these fundamental, these 1950s classic questions of ecology that are really, really hard to address empirically. But with a better toolkit and with some of these controlled, or not really controlled, but quasi-natural large-scale experiments, we can start to pick apart with more mechanistic clarity.
Yeah, very interesting. Along these lines, then, Rob, we have a question from Elodie here. What are the steps to reintroducing a species, and how do you select how many to reintroduce and when?
That's also a very good question, and I'm not the best. So when I first came to Gorongosa in 2010, I formed opinions about what I would do if I could do anything, you know, if I were in charge. And then quickly learned that as much as, you know, the best laid plans or theoretical ideas might lead you to a certain conclusion about that, the reality is you're going to be constrained by various things, logistical and political. And so, you know, zebras, right? I thought my first reaction, like literally day one in Gorongosa, and I mentioned this in an obscure article that I wrote in 2012, was I think we need zebras. Everybody, we need zebras. Okay. We've been trying to get them, but politically and perhaps biologically too. I don't want to put my foot in it. I don't know who's out there watching, but there's a notion, not firmly scientifically established, let me put it that way, that the Gorongosa, the zebra population indigenous to Gorongosa is a distinct subspecies. It does look a bit different. And anyway, the point being that you can't get zebras, you can't go to South Africa and get 1,000 zebras from Kruger, even if you wanted to, because there are, you know, the attitudes around the issue are that we want to repatriate with the breed that was here historically, and so there's only a few places you can get them from, and we try to do that, and it's, you know, and it's Zimbabwe won't let us, or it fell through, or, you know, similar thing has happened with leopards. I mean, I think, you know, wild dogs coming before leopards was partly a chance event that happened when leopard export fell through, right? So I'm not answering your question really, although I am, you know, I think one could devise a, you know, sort of biologically based, all right, I'll say something that might seem trivial, but it's not. Start with herbivores and then carnivores. And one reason that's not totally trivial is the story that I didn't get a chance to tell, which is that this is an invasive plant, horrible invasive plant, Mimosa pigra, that everyone was really worried about in Gorongosa. This pattern that I described of all herbivores converging in their diets, right? They're all going for the most nutritious foods. When they're overlapping, it's because they're eating the really good stuff. And this mimosa is like it's literally the #1 food. It's 70% of bushbuck diets in the floodplain. It's 20 to 60% of the diet for waterbuck, reedbuck, impala, oribi, and buffalo. Warthog don't eat it, but they don't need to. And so in some ways, like letting these herbivore populations build up to the point where they're actually straining the resource base is actually performed a service functionally that was useful for management. Mark Stolmans told me he really felt this was actually, you know, genuinely useful result for them to have, and I think it wasn't super premeditated, but it did. Like if predators had come in right at the jump, or let's say in 2012, right, we might not have gotten this outcome, and the predator introductions might have failed. So I think the only good answer to your question I can give, not really being my wheelhouse, is that you let herbivore populations build up, you know, even to the point where people are starting to talk about overpopulated, which is a term that a lot of people throw around. But you know, as we've seen with waterbuck, density dependence tends to take care of itself. Sorry for that rambling and slightly digressive answer to your question, but that's the best I can do.
No, thank you very much. Not rambling at all. I think we have a couple of more questions that maybe we go through from the lab, not to make this very long.
No, it's fine.
So one question we have here, and changing topic slightly, is if you think there is any place for sustainable hunting, assuming there is such, I mean, the person asking says that she or he or they don't know much about the topic. So assuming there is something as sustainable hunting, do you think there is any place of having this, for example, for waterbuck, given the very high abundance in Gorongosa? Could selective reduction of waterbuck reduce competition for other recovering species?
That's a great question. I mean, the last part of that question is a great question ecologically. And actually, if someone did decide to come harvest waterbuck, there's a lot we could learn from that. Just like, you know, any other perturbation, right? That the responses of the rest of the community to that event would be very telling. And we'd also like to get our hands on samples that we could collect from such waterbuck. So if anyone's thinking about doing that, let me know. I mean, I think the first part of the question is more of a values question, and my personal opinion is absolutely, and that could, and I think there are people in the Gorongosa project who share the view that sustainable wildlife farming for meat and protein would be a very good potential synergy between the park and the communities. So you know, speaking as an individual, I have no problem with that.
Mm-hmm. Yeah, interesting. I mean, we already, the park already is giving a certain number of waterbuck right to other parks in Africa. So that would be, it's another strategy, even though you are not going to give thousands and thousands of animals at once. Logistically is a mega operation, I imagine, right?
Mm-hmm.
Renee?
Yeah, we have a question here, Rob, from Dan. Do we know yet how climate change is impacting the habitats and species of Gorongosa? And are there any long-term patterns emerging for how wildlife is responding to ecological pressures in relation to this?
I will tell you the very few things that I know about it. One is that if you go to the chapter 12 of the most recent IPCC report and look at the maps they show there on the sort of multi-model forecasts globally, Mozambique seems to get is predicted to get the worst of the worst. So they're more longer stretches without rain, but more intense pulses of rain, so that's a recipe for landslides and flooding and all kinds of stuff. High, you know, a large degree of temperature change. So Mozambique is very much in the eye of this climate storm, and so it should be impacted. We have not really done anything to yet to measure that, except a project led by Hallie and Justine, along with everybody else here in different capacities, on the impacts of Cyclone Idai ecologically. So big cyclone, one of the most powerful on record in the Southern Hemisphere. And we had a lot of collared animals out at the time that we can, you know, look at how they responded. So, you know, insofar as that's a climate-related event that may foreshadow the future of more intense storms, et cetera, we have some information on that. And the short story there was that there was very strong acute impacts, but it's not clear that there were really sort of super profound long-term impacts, although it may be an inflection point, for example, where waterbuck decline for the first time between 2018 and 2020, possibly having to do with the cyclone impacts. We have, you know, Ryan is leading an effort. This bushbuck to Nyala to Kudu system is great for understanding thermal physiology, and that's Ryan's specialty. And so he's got a manuscript that's been in preparation since he was a postdoc with me, in, you know, almost 10 years ago, about forecasting the climate impacts on, you know, you know how these things go. It's like you got a job and then you had students in, yes. But it's going to get done. And, you know, large-bodied animals have more trouble dissipating heat than small-bodied animals, and so that makes predictions about where they should hang out and what in relation to shade and so forth. So there are these kind of projects that we're working on, but I don't really have any great answers to the question of, you know, what the signatures of climate change thus far have been.
Thank you very much. And we have to have a question about baboons. So we have a question here from Yana, and she's asking, do you think we may see a crash in baboon population similar to the crash in waterbuck and warhog crashes seen at Lake Nakuru once larger carnivore populations and other species continue to recover?
Maybe you guys tell me. I can't come here and tell you guys anything about baboons. I don't know. What do you think? I'm curious.
I don't know. I think it's very unlikely given the population you have at the moment. I think it's most likely the largest in Africa. It's just the baboons never had the same crash as the other mammals, and they recovered so well, and they are so healthy and so free from predation that I think the way carnivores recover and the way, I think the rate of recovery for the carnivores, it's going to be difficult that you get that fast enough to vaguely affect the numbers of baboons. I think other things can affect the numbers of baboons, but not just, so I think a composite of factors could affect the numbers of baboons, but not just the carnivore presence.
Right. Yeah. One thing to notice, one thing I guess I think about as you say that is, in general, omnivores have a great advantage, right? They're dietarily, they're flexible.
Mm-hmm.
Also, baboons are smart. They live in groups. They can climb. They have a lot of assets that these hoofed animals do not. So it would make sense. What is if it would make sense to me that they might not exhibit those kind of catastrophic decline?
I think in a way, for the baboon, of course, the main predator would be the leopard, and that's something that you don't envision, having very large numbers present in the park. We wish that was the case, and I think this has quite some variation across sites actually. From what I can see in Gorongosa, lions don't really go for baboons, for example, while in other sites they may go for baboons once in a while. So I think that is also the preference, what prey is available, and with this abundance of other species, so it would have to be something that would affect also the availability of prey for baboons to become the main target of these other carnivores.
What is the current population to your best guess?
The best guess is in the number of troops, so about 220 troops in the park. Now, a troop can go from 30 to 200 individuals, but they have about 15,000 baboons in the park.
Wow. Yeah, wow.
It's pretty high.
Awesome.
Yeah,
awesome-ish.
It seems to be one of the highest densities and population numbers of baboons in Africa, really. It's just
certainly the waterbuck are by far, I didn't really kind of get into it and talk because of not wanting to digress and lose time, but this density of 81 individuals per square kilometer is truly, for a 200 greater than 200 kilogram antelope, that is ridiculous. And yeah, we found in the literature people talking about 30 individuals per square kilometer as extraordinary high, the highest in Africa. So it's impressive the densities that some of these things have reached in this kind of what I think is a sort of a slow transient phase of settling into some different.
Thank you so much. And I think we end with your own question. Renee, you had a question too, and that may be our last one.
I would like to keep asking, but yeah, we do have to come to an end soon. I had a couple of perhaps related questions, Rob. One has to do with the fact that those of us who study paleoecology, we tend to think of essentially all modern African ecosystems and parks and so on as have disturbance in the past. Some very significant, some in historical time, the rinderpest in the Serengeti and so on. So in a way, thinking of any one ecosystem today as being having little disturbance may be a bit of an illusion. And the question leads to your lab trying to understand some of the basic principles, organizing principles of savanna ecosystems. How does that play into the way that we think about a place like Gorongosa? And if somewhat related to that is how would these basic organizing principles differ if you were to be doing similar research in ecosystems like a rainforest, for example? So these are somewhat questions, but trying to get at how we approach the question of basic ecological principles that your lab has been working on.
Yeah, great question and great point. I think with respect to the first question, absolutely right. Like in deep time, all of these systems have had massive disturbances, and I think the what I'm talking about really is the quasi-experimental nature of a pulse. So if in Kenya since 2008, we've run an experiment where we manipulate the densities of large mammalian herbivores in plots. And this is not so dissimilar to that, right? So you're sort of watching something. So it's not that Gorongosa is unique in having been disturbed, or even unique in obviously in terms of the conflict history, but rather that we're around to watch this one, and it's an opportunity to understand what has happened over several decades when large mammal abundances were very low, and also to kind of track the progression of the subsequent dynamics, in the way that we talked about in the talk. So I mean, I think that is an important perspective to keep in mind, and I didn't really, or I'm not really thinking about it in this way at this time. But I mean, I mean, Gorongosa could also be a model for kind of for studying disturbance itself, or the impacts of disturbance itself, which is ubiquitous and has been powerful in shaping the patterns that we see around us all the time. So completely agree, and that's anyway how my outlook on it. And then the generality, how far do these things? I'm thinking of being general, at least insofar as within the biome in question, and we have these the last remaining assemblages of large mammalian herbivores are in African savannas, and so I'm trying to understand the rules that structure those systems. Now, forest, totally different situation. So much of the plant biomass is inedible, either because it is, for at least for ungulates and for a lot of other species, either because it's wood, or because it's way up in the sky. And right, so that in a very different. I mean, I think that I do believe that the patterns that we're seeing as general sort of resource partitioning patterns are ubiquitous, and I think that the DNA metabarcoding is. There have been, I mean, a lot of studies from a lot of different groups. Elizabeth Clare's group and on bats. A recent study from there's a Chinese group was doing stuff on large carnivores, and basically when you go and look with these tools, you find that there are no two species differ in the species that they eat, and irrespective of how they differ in other regards. And the generality of that. And the mounting number of examples tells me that this is something that is real and important, in the Himalayas, in the Amazon, in Gorongosa and Serengeti. So I do think that there is some generality here that also transcends the savanna biome, but I'm not going to try to push those arguments quite that far. I'm really, focused thinking about large mammalian herbivores, ungulates. I think it's, of course, I don't know. From a paleo perspective, you want to go and say, well, what about Pleistocene Europe, and what about Pleistocene North America, and even Pleistocene Africa, where you had, you know, very different assemblages, bigger bodied and fewer ruminants. And I find all this stuff fascinating, absolutely fascinating. But it's a little that hind casting I find challenging, right? The granularity of the information I work with is tough. Although there is stuff, you know, the stuff that Willerslave's group and others are reconstructing on the mammoth steppe using similar techniques and similarly showing that diets are very different than, you know, mammoth diets were different than everyone has been assuming, and that forbs were really important. And some of these themes also, we see that same pattern as well. The importance of forbs. Forbs are this overlooked plant type that seem to have been really important in paleo steppe ecosystems and in contemporary savanna ecosystems. So there's some, I think, some generality possibly going back in time and some generality across systems, but primarily I'm thinking about, can we understand what's going on in contemporary savannas, just from as a matter of personal preference.
Thank you so much, Rob. This is, well, I think I could continue talking for another couple of hours.
I would love to do that, but maybe when we're, are you going to be, anyway, we can talk about it some other time.
I think we would have a lot to discuss, and I'm having ideas here about, actually, maybe there are some things we can say about our fossil records following, you know, the principles and what you are doing, some things that may be assumed or extrapolated. So there are a lot of interesting potential connections definitely there.
I would love to talk about that, yeah.
We will, I hope so. And I on behalf of the lab and the department and the University of Oxford, we thank you again enormously for your time, for this brilliant talk, for your willingness to show us and tell us about all these different bits of research that you and your team have been doing. We really hope to catch up soon about more of the research in Gorongosa and elsewhere. And I'm going to close this session today. I'm going to thank the audience that kept with us until now, and I'm going to invite everyone to come back next week, same time, to attend a talk that is going to be again on Gorongosa, but we are going to listen about a multi-level approach to Gorongosa baboons Landscapes of Fear by Philippa Hammond. So please join us next week. And thank you everyone for attending today. Bye-bye now.
Thanks everybody.
Bye-bye.
Thank you, Rob.
Thank you, Rob.